Well, welcome to our next session. So Franca Fraternali from King's College in London is going to talk to us about genetic variants.
Hello, everybody. Good afternoon to everybody, and I'm glad to meet you virtually in this experiment that we are going to perform today. So I will first give a webinar and with a presentation of the background to what will be your practical session later. And so the first 45 minutes are a kind of a lecture-like and background for the exercise and for your introduction to the subject. And please post questions. There will be a question and answer session at the end. So we will have about 10 minutes for that. So I'll just start by sharing my screen. And I'll start by opening the presentation. Okay, so the material presented here has been assembled it's largely based on the research of my group, and most of the material of the tutorial is being assembled by Joseph NG, who is a postdoc in my lab, and you will have assistance by Molly Gravett and Timir Weston during the sections of the exercise. You will see sometimes, because some of the concept or the slides I have, some are more of introductory concepts, but some are more presenting more difficult or concepts or formulas. You will see this sign posting in the slides when the concepts are more complex, and I won't go totally in the details of the concept I introduce, but you will have for reference the formulas, and you can anyway email me later if you're interested in some of these specific concepts. So let's start by saying that in the past 5,000 years, the population has acquired an enormous amount of genetic mutations, and these genetic mutations have arisen within each generation. So humans have an abundance of rare genetic variants, and many of those are in the protein coding genes. Of these single nucleotide variants, 70% about arose in the last 5,000 years, and on average, a percentage of them were potentially harmful, and 86% of these arose in the past 5,000 years. So this means that most diseases are caused by more than one variant, and that disease can operate through different mechanisms and genetic pathways, and so they can have a different pathway or course in different people. So if we look at one of the concepts I'm going to introduce today, is just, if you look at this type of diagram, which represents the variant penetrance, that means which is proportional to the number of individuals that present a specific mutation versus the disease type. So you can have diseases that are very rare, uncommon, or common, versus the variant penetrance that goes from low to high. In this kind of diagram, you can see that there are some diseases that are very different to identify genetically, so it's difficult. There are Mendelian diseases that are the ones that are genetically identified, and there are some low frequency variants that have an intermediate penetrance. So all these scenarios are possible, and there are and the common disease on the other end are mostly identified by genome-wide association studies. So the concept here are, and I give to you, it's what is a variant. So it is a unique protein or DNA sequence that has been generated by mutagenesis from the wild type. The penetrance, I just mentioned a few minutes ago. The allele frequencies, is the relative frequency of the variant of a gene at a particular in a particular population, and this is expressed generally as a fraction. And some nomenclature that you may have heard independently from your background is single nucleotide polymorphism. That is a single nucleotide, a substitution of single nucleotide at a specific position that is present in a considerably large fraction of the population. Instead, if you do not consider this frequency limitation, we are talking generally about single nucleotide variants. So why harmful variants are fixed in the population? You could ask why, why nature doesn't defend itself by this. And one example is the sickle cell anemia. I'll give you two examples why we think there are good reasons to think why these mutations are fixed in the population. In the sickle cell anemia, you have a single point mutation of a particular position where a glutamate 6 is mutated in valine in the oxy and deoxy form. So you can see that this by changing this mutation, by the introduction of the mutation has some consequences on the structure, which is represented here. And this is the heme group with the oxygen binding site, but the dramatic macroscopic changes are expressed as are manifested in the red blood cell shape. In fact, the oxygenated form of the hemoglobin stick to each other, and they produce these stiff fibers that are not the classical red blood cells. And the malaria parasite cannot live well in these stiff fibers. So the parasite that cause the tropical disease malaria, they spend most of the time in these red blood cells, and somehow they not being able to live in this blood cell confer a certain resistance to the people that catch malaria. So in fact, if you go and look at the distribution of the sickle cell anemia trait and the geographical distribution of the malaria, you see an incredible superimposition of in Africa. This is mostly in Africa, but also in the Mediterranean area, we had large infestation of malaria. So this tells you that a potentially deadly mutation is acquired in the population as a defense against a third event, in this case, the malaria malaria disease. In the cystic fibrosis, again, we have a single amino acid deletion, and this is actually is a deletion of phenylalanine, and this causes the channel blocking in its mutated form, and therefore the individual with this particular disease they have a ionic imbalance, and they have mucus and thick mucus and problems with the pancreas. So why this mutation was fixated? There are different putative reasons that have come across. The fact that that there were cholera spread around the world in the beginning of last century, the typhoid fever, and some kind of resistance to tuberculosis. So there are reasons why certain pathogenic mutations are fixated in the population. We want to understand the origin and the consequences of mutations, and we want to look at them from the point of view of how do they occur on the DNA, and what causes the mutation at the DNA level in the product of the DNA, or and more specifically, we will focus on the impact of this variation at the protein level. So we know from the central dogma of molecular biology that the information that is transferred from the DNA to the RNA to the protein, this is a process that produces or passes information at the different steps, but the information that is in the code stops at the protein level, and at the protein level you have proteins that act alone. So they are formed, they are folded, and they are act alone or in complex with other cellular proteins in vivo, or they can assemble to perform a certain particular function. So what happens to the mutators that leave signature on the DNA? So the DNA, we know that our DNA is constantly mutated. So they are mutated by certain mutagenic agents. One of them is the ApoB protein. These are enzymes that leave a mutational signature onto the DNA. And has been seen in recent years, incredible work has been invested in this, in characterizing this signature. And we know that there are particular patterns on our DNA that are really typical of aging, or for example, of smoking. The smoking signature is clearly evident. So we can read our DNA and know actually what have we done in the last few years of our life. So, but why we want to go to the structured level? So we go from the DNA to the protein, from the protein sequence to the structure. And this is because knowing the structure is essential to know a lot of information about the protein. First of all, you can extract the multimeric state by knowing the structure. You could look at ligand and functional states. You can look at the surface proteins and extract putative binding site. And you can look at the evolutionary relationships. So one example that clarifies and is widely known is P53. And we will look at P53 from the point of view of the 3D structure. This is a tumor suppressor, and it's involved in a number of functional interaction with a number of genes that control the cell cycle arrest. So also other function like DNA repair and apoptosis and so on. So the P53 is a folded central DNA binding domain, which we call DBD, and act as a tetramer in vivo. So if you look at, mutation in the DNA binding domain is found in 50% of all human cancer. So it's incredible, rich and accruing mutation across the board of different types of cancers. In fact, if we look at the worldwide distribution of cancers and P53 mutation in the developing countries and developed countries, we see that the vast majority of this cancer is attributable to small mutation into the PDB gene. So this is a three-dimensional structure of one monomer. It acts as a tetramer, we said. Here is a dimer. So there are the loops in this region, and the loops are at the interface, and there is a DNA binding at this interface. Most of the mutations here, if you look at this pink area here, this is the accumulation of the mutation, the non-mutation in the specific positions on P53 DNA binding domain. We can see that these are some of them, if we go in the specific in characterizing them, some are characterized as contact, either directly in contact with the DNA, and some are structural residues, which mutation results in perturb the structure or results in contact loss of this direct contact or mutation. So this is clear that the structure explains to us why this mutating these particular residues, we lose the interaction, the vital interaction with the DNA in this case. People have tried to combine different data to extract some significant, some reason of this mutation, for this mutation to become so detrimental for the molecule. But already by mapping this as a number of variables, like calculating the energy, looking at the conservation, looking at the loss of activity, we see that if we map this type of phenotypic outcome or calculated outcome onto the structure, we see that none of them explain exactly, none of them shows the same type of scenario. So we need to merge data and to have a number of data that can explain different aspects of the possible reasons of perturbation due to the perturbation or mutation onto the structure of the protein. There are also rescue mutation. It is a very important concept because especially with P53, there are some mutants that can be restored by a second site suppressor mutations. So you can restore the activity of the protein or the function of the protein. And this is very important because some therapeutic drug may rescue mutant P53 with this mutant. So if we understand this mechanism of rescuing, it's important. We have done in collaboration, previous collaboration work in trying to understand or trying to identify rescue mutation. In the lab of my lab and Arianna Fornilli, we developed the method DFS to detect rescue sites in a protein. And this is the link to the software. And we applied single force or double force either across the entire protein, and tried to use the double force to rescue the mutations. And so we came up with the rescuability index, and we mapped this rescuability index on the protein, and we have a very good agreement with experimentally extracted rescue mutants. And particularly for a special region in the protein that contains a lot of pathogenic mutants, rescuing the activity of the protein. So, but I want to show you some challenges in this variant mapping. You can think about, okay, I have a protein, I have the sequence, I have the fold, and then I simply map the mutation at the position of the fold. I look at the contacts, I look at the energy, and so on. For certain proteins, this is very difficult. Because of course we have a structure sequence gap. We don't know the structure of all this protein, expressed protein in our body, but also some proteins are specifically complex because they are very large and they have a complex substructure. So Titin is the largest protein in our body. And a few years ago, people came up with the idea that Titin is a major human disease gene. But in fact, due to the large size of Titin, the majority of healthy individuals possess rare Titin missense variants. So if you look at an healthy individual, you have mutation in Titin. So this results in the paradox that rare Titin variants are in fact commonly found. So we try to tackle the problem because the rare Titin missense variant has been associated with disease, and particularly here I talk about cardiomyopathy, and particularly in hypertrophic cardiomyopathy. In this case, you can see that in this myopathy, the wall of the heart becomes thickened. But there are many other diseases related to muscle and cardiac muscle that involve Titin. So it was important for us to understand. The structure of the muscle is complex. We have different in the sarcomere, different bands, and they are elastically moving with respect to each other. And Titin, this is a representation of Titin. You see in green the Titin substructure. These are all the subdomain of Titin here represented in green as pebbles. And we see that, I mean, the number of amino acids is impossibly large to treat as a single protein. So we have to split the problem in their domains and then constituting domains. So we can have the structural information of the single constituting domain and see what happens to this by mapping mutation onto this. This resulted in a large-scale effort in modeling all these domains. I won't go again in detail of this plot, but I want to tell you that if you look at this red bar, which are proportional to a better quality of the model, we modeled all these domains and we increased the model quality of previously available models and made some of the homologs that have a structure very close in quality. So we restructured, basically we remodeled all the ultrastructure of Titin. And we wanted to look at variants that may present pathogenic Titin variants. There are some cases in which you have combined impact of two distinct variants. And we want to focus at the molecular level, as I said before. So what we did, we decided to prioritize variants using in silico techniques, and we generated a database which we call TitinDB, in which we did map all the non-synonymous NIPs known in the population and in the one associated with disease. We assessed with computational tools the impact of this on the molecule. We had all the homology domains and we mapped this on the structure. With the software to predict the impact of mutation, which you will use or which you will see the outcome today, is based mostly, the older software are based on sequence and conservation. Some contain secondary structure and structure-derived properties like polyphen 2, and some are really tailored to structure 3D structure of proteins and thermodynamic stability, and this is MCSM. You will use this software today. Again, very, very quickly, because this is a concept that requires a seminar in itself, but the method you are going to use is called MCSM, and we use this largely in our lab. Is from the group of Tom Blandell and Douglas Pires is the developer, and it basically contains geometrical feature inputs and physicochemical features inputs. So it calculates not only neighboring and the topology of the protein, but also taking in account of physicochemical feature of we have the structure, we input the structure of the wild type, and it calculates the impact of the mutation within this structure. So this can use some local environment, physicochemical properties, and has been trained on a data set of single point mutation that have experimental free energy changes. With this software, we can assess protein stability, protein-protein affinity, and protein nucleic acid affinity. So it's very useful for the assessment of at the structure level of mutation impact. So we know that in vitro, one can calculate saturation mutagenesis. What does it mean? I can change every amino acid in my structure in every position in my protein in every other amino acid, and I can have some form a phenotypic output reading that tells me how much this change has affected the protein. And so you can generate some kind of landscapes in which you know which are the position within your sequence, within your structure or sequence that are more delicate or are more susceptible to mutation. And this is important because you want to calculate the impact of the mutation, but this is really expanding the landscape of calculation enormously. But it would tell you exactly how each amino acid within the protein can contribute or can be damaged by the perturbation with the mutation. And this can be done, this is done of course with the cell functional assays experimentally. And one example that many of you may know is alanine scanning, but this can be done also at the computational level. We performed with MCSM and in silico saturation mutagenesis of all the Titin modules, and we have reported the impact of these mutations, large-scale variation and impact within the Titin database. This is a database, I don't think we will use it today. I give you the references, but I show you in brief which information you find. You can have you can have a table with all the mutation in the common population and the residue impact or the frequency of the position, and so you can map the mutation interactively and look at the position of the mutation onto the structure, and you can also see what is the so we can rotate the structure, and you can see that, for example, in this specific case, pathogenic mutations are all clustered in one particular side of the protein. And then you can also look at the reference in the literature. I have a lot of information. So these are things that you can explore with this software. In the lab, we look also at protein-protein interaction, and so we try to see also genetic variation. How is it affecting the neighboring in a more coarse-grained manner, because we are not looking now only at proteins as isolated object, but we look at proteins in the context of their interaction in protein-protein interaction network. But of course, it can happen that protein can disrupt the protein core, they can disrupt interaction sites, and they can affect multiple interaction with multiple proteins, so it can impair multiple interaction, and some other mutation can be really close to functional sites. So there is a lot of different scenarios that one can consider in evaluating this. We have decided to look at the protein in terms of anatomy of the protein. What do I mean by that? I have a protein, I have the sequence. I can look in terms of domains that are formed within the protein, so single folded unit, and I can look at their structure. This can map into structure, and can look at core, surface, or interface region, or I can look at the domain type or a functional site and protein regions in general. I can output protein region statistics. So we have embarked in this large scale of all the human protein encoded genes, and we have mapped onto these genetic variants from germline genetic disorder, from cancer genome, somatic disorder, rare population variation, and common population. Ideally, you would like to see a difference in this in the location, or you would like to understand if this variability can be explained by looking at the location on on the protein structure or the function of the proteins and the pathways that the protein is subject to. So we looked, in fact, at biological pathway, structural localization, and proteomic features. And we thought, okay, could this be considered a proxy for their pathogenic, or could have information about their potential pathogenicity? So can they be discriminator between variants and their impact? So we came up with the first general outcome that the missense variants target distinct functional pathways. So what we did, we took the protein, we mapped the variants to proteins, we mapped to the pathways that they are involved with. We looked at the enrichment, so which pathway are mostly populated by these variants, and we look at clustering of this pathway. One very interesting outcome was the first that the variants that we observed were localizing in 3 different type of pathway functional clusters. The pathways were first clustered according to three type of clusters: the proliferation pathway, nucleotide processing and response, due to the pathway enrichment. And interestingly enough, we saw that if we project in terms of protein PCA, we see that the pathway, the common and rare variants, are completely in a different subspace with respect to the clean bar and the cause. These are pathogenic and these are non-pathogenic. The common and rare variants, if we look at in a 3D cluster, we can see that the common and rare variants are within the green cluster, which is the one that is called response, which is due to the olfactory, drug metabolism, some very, very basic functions of the protein. And the pathogenic ones, clean bar and cosmic, are in between these other 2 clusters. One is the proliferation, which makes sense that the cancer variants are within this pathway, and the other one is a very essential to the cell pathway, which is the ribosome, the ubiquitin proteolysis, and the spliceosome. So I think we can see that this mutation targets specific pathways. And so there is some information in these variants that we can use in terms of predicting the impact that they can have. I won't go in the detail of how we calculated this enrichment score, but what we looked in essence, we looked at the proteins, and we look at how many proteins within this type of scoring function, which is called the binomial cumulative distributed function, are enriched in missense variants or depleted. So we have a way to monitor those. And then we looked at what happens at the interface, core, or surface of protein structure. And we have the categories of GNOME. This means common disease, common variation, cosmic, cancer-related, and clean bar. Clean bar is really variation determined in the clinic, so it's really pathogenic. These are also pathogenic, as we know. We expect no pathogenicity here in the common, maybe some pathogenicity in the rare mutation, possibly. But we can see that actually they are the variation, the pathogenic variation is depleted at the surface, enriched at the interaction core, and you can see that the common variation is it's really depleted in this region. So we see a different scenario that seems to be meaningful, of where and how a specific mutation goes and localize on a specific region of the protein. If we look at other more large-scale data on proteins and protein network, protein interactions, we can look at the stability of the proteins. And so we have large-scale data from profiling experiments in thermostability of the protein, and we can do the same exercise, look at the enrichment of variants in these proteins, and we see that you see an enrichment in the protein for the stable protein, and a depletion of the pathogenicity variation at the core. And so it's for the abundance of the protein. So in the cell, of course, certain proteins are more abundant than others. And of course, their abundance must play a role in the regulatory fine-tuning of the pathogenic mutations. And we see also a clear discrimination between the common and the pathogenic variants. So there is a distinction between the structural impact and the process that this mutation occur. Target. But of course, we have already decided in the beginning which are common, we have a data set that we have given to our analysis of the impact. So therefore, we need to validate this with an external validation tool. So suppose we do not know which is the type of mutation we are considering. So we put all our mutation in a one pot, and we score them with an external predictor. And we want to see if we see this scenario that I have shown to you before as reproduced by this external, by this unique plot that can they can distinguish between the different variations. So we did the exercise with one tool that is called Revel. Revel is a very recent package, and we scored all this pot of variation, and we saw that then if you, because you know which one they are later on, you can go back and we see in fact that most of the tolerable are common, and you see the clean bar are mostly damaging. So this, what we observed as phenotypic of features that we observed are really reproducible in terms of what is the role in the impact. And so we did also this for other type of data in which we had cellular data. So, but it's just to say that we could reproduce the results that we had from the beginning when we had the data set split in different categories. So in general, what we come up with, the outcome of all this analysis can give us some molecular feature of human proteins that are important in terms of disease-associated variants. So they are enriched at the core and interacting interface, are depleted at the surface, and the population variants are enriched at the surface. They are enriched in the abundant protein and in the stable protein, and they are sensitive, therefore, to missense variant. And these are the ones that are resilient to missense variant. Of course, we don't know anything about the history, doesn't tell us anything about the variants that have disappeared in the population. This is a difficult prediction to implement. So we have a database that on which you can look at this variant enrichment at the protein level. You need to know the protein uniprot protein. You will learn what this means today. You will use this, and you can look at what is the variation in these proteins, and so you can look at where do they occur, and in which pathways they are enriched. You will also use this software developed in the lab, which is called PinSNPs. This is really a visual and quick outcome of a precompiled database. So it's a bit not completely updated. We will merge soon these two tools that you will use today. But in this database, you will look at the protein, a singular protein, and the protein interacting. So you'll know the neighbors and interactions, and you will look at the interface mutation of this particular databases. These are now a bit old, but you have in ZoomVar the most recent version of this variation, and you can look at the protein binding mechanism. You will have plots like this. You will generate plots like this in which you can look at a specific protein. And for example, this is a complex. I had here as an example a complex because you can see the interface between the two proteins here. You can map variants that occur at the functional side. They are the blue one, and the cosmic SNPs are in red, and the post-translation modification. So you can annotate all the information that is in the database precompiled, but you can explore these in the 3D structure, in a 3D structure inspection view. So this is PIN-SNPs based on a number of interactions resolved and variants, and this is the work we have performed on that. I think I like to stop here with my background. I'm sorry if many of these concepts are very complex, but I think I wanted to give you reference and information about most of the things we're going to touch upon today. And I need to thank the people who did the work. It's, Anna, and historically Julia and Herrera Braga and Grace did PIN-SNPs. Anna took the ZoomVar and the Titin, and Joseph completed the ZoomVar project, and is also very much into cancer mutation. So he is more dealing with the cancer variants. And of course, there are collaborators and funding agency, and I thank you for your attention. And I'm ready to